Hello, my name is Cindy Atwell, and I'm the head of business development at Precision BioSciences. We're a clinical stage biotechnology company leveraging a next generation genome editing technology for the development of human therapeutics. It's been an exciting year at Precision. We've progressed our allogeneic cell therapy platform, initiating a stealth cell CD19 clinical study earlier this year, as well as a cohort studying a combination of our BCMA product with a GSI inhibitor. On the in vivo gene correction side, we announced that we anticipate filing 3 CTAs or INDs over the course of the next 3 years. We continue to advance our programs with our partner Lilly, including DMD, a liver target, and a CNS target. And we recently announced a deal with iECure to apply ARCUS for gene insertion in 4 orphan diseases, including PKU and OTC. Precision BioSciences is a public company, and I will be making forward-looking statements today. Please refer to the disclosures and risk factors in our SEC filings. ARCUS was invented at Precision and was developed with an eye towards clinical translation without unintended changes to the genome. We're focused on addressing serious diseases through unlocking the full potential of our ARCUS genome editing technology. We're seeking to apply ARCUS to cure rare diseases, making a permanent change to patients' DNA. We're seeking to overcome cancer through the development of allogeneic CAR T therapies. We chose to invest early in manufacturing as we see this as a strategic advantage. And we've established GMP allogeneic cell therapy manufacturing as a core capability. In July 2019, we opened MCAT in RTP, a fully GMP compliant facility. We're able to produce all the key components across the portfolio for CAR T and in vivo gene correction therapies. And we're currently producing clinical trial material for our CAR T programs. Our CAR T pipeline includes 3 first-generation CAR T products in the clinic, as well as a second generation, the PBCAR19B program, which entered the clinic in the first half of this year. Also in the first half of this year, we initiated dosing of a combination study with our BCMA CAR T product and a GSI through partnership with SpringWorks. Additionally, we anticipate filing an IND for the BCMA stealth program in 2022. Part of the reason we're able to rapidly advance our cell therapy programs to the clinic is that there are a lot of similarities across the 3 therapeutics that we have put into the clinic. Namely, that we use a proprietary one-step gene editing approach to insert the CAR into the TRAC locus. We simply switch out the gene inserted, which is the CAR, targeting antigens relevant to the type of tumor we are aiming to treat. This allows Precision to rapidly apply its learnings across our cell therapy pipeline, as well as to rapidly develop new off-the-shelf CAR T therapeutics. Our lead program is the PBCAR0191 allogeneic CD19 CAR T product. And we're currently testing this pipeline product for the treatment of relapsed refractory NHL and relapsed refractory BALL. The primary objective of this phase 1/2 study is to assess safety and maximum tolerated dose. Secondary objectives include clinical anti-tumor activity, and we are also looking at the expansion, trafficking, and persistence of our allogeneic CAR T cells. Importantly, we are exploring approaches to drive the durability of clinical activity. This includes exploring opportunities to enhance allogeneic cell expansion, trafficking, and persistence, such as enhanced lymphodepletion or novel lymphodepletion approaches. This slide describes the enhanced lymphodepletion patient cohort. Patients were heavily pretreated and had aggressive disease. Over 80% of the subjects had aggressive lymphomas. The median line and mean line of therapy was 7. 4 subjects had prior CD19 CAR T therapy. And importantly, median time from eligibility confirmation to start of lymphodepletion was 1 day, validating the potential of PBCAR0191 as an off-the-shelf product. This slide shows the safety profile in standard lymphodepletion and enhanced lymphodepletion patients. Importantly, there was no grade 3 or higher CRS. Only one patient had a grade 3 ICANS in the enhanced lymphodepletion patient population, which resolved within 48 hours of treatment. Importantly, there was no graft versus host disease. As anticipated, with the enhanced lymphodepletion, we do see an increase in grade 3 neutropenia and increased infection rates. Now, we achieved the goal with the enhanced lymphodepletion cohort of increasing our Cmax and cell persistence. We saw a 56x higher Cmax, as well as increased persistence by flow, as well as PCR, as compared to the standard lymphodepletion cohort. This resulted in increased response rates and complete response rates. The overall response rate for the enhanced lymphodepletion cohort was 75%, with 75% overall response rate for the CAR-T naive patients, as well as patients who had received prior autologous CAR-T therapy. Complete response rates were 50%, with the same percentage of patients achieving complete response in the naive and prior autologous CAR-T patient populations. We are also pursuing clinical development of our second generation CD19 product. This is our CD19 stem cell candidate, PBCAR19B, which includes immunoevasive technology and is shown to delay both T cell and NK cell mediated allogeneic rejection in vitro. We believe that delayed T cell and NK cell mediated allogeneic rejection is likely to result in improved persistence of allogeneic CAR-T cells. The IND was accepted by the FDA, and we dosed the first patient earlier this year. Let's now turn to ARCUS for use as an in vivo gene editing tool. ARCUS is precise and versatile. The impact of precision is safety and specificity. The impact of versatility is that ARCUS is easy to deliver to the liver as well as other types of tissues. Additionally, ARCUS can perform complex edits for gene insertion and repair. When developing ARCUS, our genome editing pioneers looked to nature for a better editing system, better specificity, and better versatility. Our team started with a natural enzyme called ICRE1, which evolved in nature specifically to perform precise gene insertion in a eukaryotic organism, algae. We developed a process to engineer ICRE1 and reprogram the enzyme to target new genetic sites. And this is what we call ARCUS. ARCUS has 4 key advantages. Safety, the enzyme has a natural off switch and doesn't edit DNA unless it's productively bound to its target. Delivery, ARCUS is only 334 amino acids long, and the small size permits delivery either by LNP or AAV. The type of edit, because of the unique type of edit, which leaves a 3' overhang, ARCUS can be applied to efficiently knock in or knock out target genes of interest. And it's proprietary. Precision has full control and freedom to operate with the ARCUS platform. Our overall strategy in gene editing has 2 steps. Step 1, we would like to achieve clinical validation using both AAV and LNP delivery with liver targets. The first programs will use a knockout approach to maximize the technical probability of success. Step 2, expanding into white spaces where other technologies may not be able to go and where ARCUS is uniquely suited. This includes complex issues and delivery to tissues outside of the liver. So if we look at our pipeline, the first program entering the clinic we anticipate will be PCSK9, using an AAV delivery. We anticipate CTA filing in 2022. This work is being partnered in collaboration with iACure. The next 2 programs we anticipate in the clinic will be PH1 and HBV in 2023 and 2024, respectively. After that are our 3 programs with Eli Lilly, DMD, an undisclosed liver target, and an undisclosed CNS target. We recently have shown compelling large animal data in other targets as well, such as TTR and ADRP, and we are seeking to partner these programs. Let's now turn to our PCSK9 program for treatment of familial hypercholesterolemia. Now, familial hypercholesterolemia is a rare genetic disorder leading to high levels of serum LDL, severe cholesterol deposits, and early onset heart disease. PCSK9 is a secreted protein which is involved in recycling of the LDL receptor. It's a well-validated target for cholesterol control. And we have been working for several years with Dr. Wilson's group at the University of Pennsylvania to demonstrate long-term stable knockout of PCSK9 in non-human primates. And Dr. Wilson's group recently published a paper in molecular therapy showing 3.5 years of stable PCSK9 knockout with an ARCUS nuclease. Now, the first generation ARCUS nuclease, described here as M1 PCSK9, we looked at 3 different doses of an AAV8 delivered ARCUS nuclease in non-human primates. And we saw a maximum of 82% reduction in PCSK9 levels. The next version, the next generation, and referred here as M2 PCSK9, recognizes that same exact target sequence, and we really focused in here on the middle dose. This is our clinical candidate, ARCUS nuclease, which will enter the clinic. Correspondingly, we saw stable reductions of LDL in the non-human primates, both with the first generation ARCUS nuclease as well as the second. Although the PCSK9 target is the liver, we are planning to use an AAV vehicle for delivery. The reason is that in this particular patient population, familial hypercholesterolemia, the LNP uptake by the liver is anticipated to be impaired due to the impaired lipid uptake in the liver with these patients, And there is no effect, such effect with AAV delivery. Turning now to our PB gene PH1 program for treatment of primary hyperoxaluria type 1. This is a rare genetic disease characterized by accumulation of calcium oxalate in the kidneys, which lead to painful kidney stones and ultimately end-stage renal disease. A combined liver-kidney double transplant is often required to treat this condition. Now, this slide shows data from our NHP study in which the ARCUS HA01 nuclease was delivered through a one-time IV administration using the AAV research vector. We recently announced that the clinical candidate will be delivered via LNP. This slide shows that the one-time treatment reduced HA01 by greater than 90%, reducing GO protein levels by greater than 80%, and increasing serum glycolate levels significantly, reducing downstream production of oxalate. This program has been prioritized by Precision, and we anticipate filing an IND or CTA in the 2023 timeframe. We are aiming to develop a functional cure for HBV. So focusing in on our HBV program, we are using a lipid nanoparticle to deliver ARCUS using a conserved site in the HBV genome. The ARCUS nuclease cuts the cccDNA as well as sites in which the ARCUS has integrated into the genome. HBV-infected primary human hepatocytes were treated with ARCUS delivered via either lentivirus or mRNA. ARCUS was found to have significant antiviral activity measured as a reduction in total cccDNA and significant indel mutations. This demonstrates the inactivation of HBV, which has integrated into the genome. The ARCUS treatment also resulted in significant reductions to HBV-secreted S antigen, a biomarker of the infection. As there is no good model for HBV for gene editing, our team developed an HBV and HP model. This is really a episomal model, and non-human primates were administered AAV carrying a partial HBV genome sequence. ARCUS was subsequently delivered as an mRNA lipid nanoparticle. Now, the liver was collected for indel and copy number analysis by PCR. Because the animals were immune competent, secreted S antigen was quickly neutralized in all animals. As such, S antigen is really not a useful biomarker in this particular model. However, we did see a significant reduction in the AAV copy number, as well as a significant number of indel mutations, really indicating an inactivation of the HBV virus for integrated DNA. Overall, we are very excited about what we've already accomplished at Precision and about what is still to come. We have achieved a number of milestones in 2021 and across our cell and gene therapy human therapeutic programs. Leveraging ARCUS, we are rapidly progressing the development of human therapeutics for serious diseases. We have already generated initial clinical data that validates our approach in allogeneic CAR-T and are advancing a portfolio of in vivo gene correction programs towards the clinic. We are well funded and have cash into 2023, which will allow us to continue to push forward to realize our vision of improving life through genome editing. Thank you for your attention today. Given the time constraints, this presentation included a subset of the information found in our full corporate deck. Please visit the Precision BioSciences Investor Relations website for the full corporate slide deck, as well as the most up-to-date information on the company and pipeline.